Good afternoon, ladies and gentlemen. Today, the topic of lecture is Enterobacteriaceae family, exactly genus Escherichia coli. Escherichia family, Enterobacteriaceae family, is a very large family. It includes many genera, Enterobacteriaceae family, and Enterobacteriaceae family bacteria, all of them are gram-negative rods, whose natural habitat is the intestinal tract of humans and animals. The Enterobacteriaceae or Enterobacteria are facultative anaerobes or aerobes, are able to ferment a wide range of carbohydrates, possess a complex antigenic structure, and are able to produce a variety of toxins and other virulence factors. Family Enterobacteriaceae have the following characteristics. Bacteria are short gram-negative rods, either motile with peritrichous flagella or non-motile, some of them. They don't need enriched media. They are able to grow on peptone or meat extract media without the addition of sodium chloride and other supplements. Just 1% peptone water is enough for them. Grow well on McConkey's agar. This is kind of differential media, and you did study last semester differential media. Those are used for diagnosis of disease caused by Enterobacteria. Grow aerobically and anaerobically, because they are facultative anaerobic bacteria. They are able to multiply in both conditions, in presence of O2 and in absence of O2. Of course, they are cultivated in presence of O2 because of it's easy for microbiologists to create aerobic conditions. They are able to ferment glucose, often with gas production, and are catalase positive, oxidase negative, and reduce nitrate to nitrate. They are biochemically active. Most of them have a lot of saccharolytic enzymes, are able to ferment many sugars, and most of them have proteolytic enzymes. They are able to destroy proteins. The Enterobacteriaceae family includes over 20 genera. Some enteric organisms, for example, Escherichia coli, are part of the normal flora and accidentally cause diseases. They belong to normal microbial flora, and they play a very important positive role in digestive tract. They participated in lymphoid tissue formation. They produce enzymes necessarily for digestive. They produce vitamins B and K group. They inhibit the multiplication of pathogenic microbes. They are participated in detoxification reactions, so they play a positive role. At the same time, I am going to remind you that all representatives of normal microbial flora, they are named conditional pathogenic bacteria. If they are able, if they have opportunity, if they are able to multiply, or maybe they go from digestive tract to by bloodstream or inside the body to internal organs that should be sterile, they are able to cause diseases too. So this is negative role of normal microbial flora. I need to remind you the positive role and negative role. So Escherichia belong to normal microbial flora, while others bacteria such as Salmonella genus, Shigella genera bacteria are regularly pathogenic for humans. And you did study the genera 2 disease in humans are caused by bacteria of the genera that belong to Enterobacteriaceae family, Escherichia genus, Shigella genus, Salmonella genus, Klebsiella, Proteus, Yersinia, and others. So more than 20 genera belong to this Enterobacteriaceae family. And now we will study bacteria of the genus Escherichia. What are they? Genus Escherichia includes several species, but the only species causing diseases in humans is Escherichia coli. Other species don't cause diseases in human beings. So diseases caused by Escherichia coli are named Escherichiosis in general. Escherichia coli properties. Morphology. They are gram-negative rods with rounded ends. They are singly arranged, very often in smears, stained by gram staining method. They are stained well by gram staining method. Microcapsule is present, motile due to perithetically arranged flagella, and don't form spores. Memorize that all bacteria of Enterobacteriaceae family, all of them are gram-negative rods and they don't form spores. Really, you have to know, you did study bacteria, pathogenic bacteria, spores forming, and they are bacteria of Bacillus genus, causative agent of Bacillus anthracis, and bacteria of Clostridium genus. Clostridium genus bacteria cause tetanus, botulism, and gas gangrene. So bacteria of two genera, pathogenic bacteria, just of two genera, are able to develop spores. Other pathogenic bacteria, pathogenic for human beings, they don't cause spores. Memorize, it's important for exam. There is photo of smear stained by gram staining, and there are Escherichia coli. They are rods thinly arranged, and they have fuchsine color, so they are gram negative. Really, they're short rods. Short rods. Then there is photo of Escherichia coli made in electron microscope, and it is possible to see well flagella. How many flagella have got this Escherichia? When you did study flagella of bacteria, I gave you information that some bacteria have just one flagellum, and they are named monotrichous. And the example was the Vibrio cholera. And some bacteria have many flagella around the cell, they are peritrichous, and the example exactly are Escherichia and Salmonella have the same flagella, not one flagellum singular, flagella plural, and Campylobacter and Helicobacter have a few flagella at the one end, Alophotrichus, and Campylobacters have flagella on both ends, and they are named Amphitrichus. So I'm repeating because of on exam, you should draw a picture of them and give an examples of bacteria. So Escherichia are an example with Peritrichus, of Peritrichus. Cultural characteristics. Like other Enterobacteria, they are facultative anaerobes. They are able to grow up in presence of O2, molecular O2, and in absence. Of course, they are cultivated if many pathogenic bacteria are facultative anaerobes. And they are cultivated like strict aerobic bacteria in presence of O2. at ordinary petri dishes, and if they are cultivated in test tubes, the test tubes are closed just by cotton stoppers, because they are able to multiply in presence of O2. They are able to grow on ordinary media, basic media, like nutrient agar, at temperature of 37 degrees by Celsius in pH 7274. Escherichia develop S colonies, S-type colonies, sometimes are really, so you should memorize typical colonies. Typical colonies of Escherichia are S-type. This S means smooth. Smooth are elevated colonies with smooth surface, circular ends, and R-type colonies are flat colonies with rough surface and with irregular ends. You have to know from last semester you did study types of colonies. They are the only 5 causative agents among pathogenic bacteria form R-type colonies, and you should memorize them, such as Bacillus anthracis, causative agent of anthrax, then Yersinia pestis, causative agent of plague, then tuberculobacteria, mycobacteria develop R-type colonies. One biovariant of Carinobacterium diphtheria, exactly biovariant gravis, and mycoplasma. So you should learn by heart these 5 causative agents R-type, they form R-type colonies on solid media, and other pathogenic bacteria have to know for sure. Typical colonies are S-type. It's important because of in diagnosis, in microbiological diagnosis, cultural method is used very often, and we are looking for suspicious colonies. Suspicious colonies are colonies of presumptive causative agent, and we have to know what colonies are formed by suspected causative agents, and we are looking for S-type colonies or R-type colonies, with color, without color. So it's important. There is a photo of colonies of Escherichia coli on nutrient agar. Small S-type colonies without color. Pay attention, Escherichia don't produce any pigments. If they grow on nutrient agar, the colonies have no color, any color. Memorize, Escherichia coli don't produce pigments. But in case of, for microbiological diagnostics of intestinal tract infections, when feces are used for inoculation, nutrient agar is not used. Differential media are used. Those content of nutrient agar plus lactose plus indicator and plus alkaline. The lactose content medium are used for differentiation, lactose positive and lactose negative bacteria. Some bacteria are able to ferment lactose, some bacteria are not able. And it is possible to differentiate colonies by biochemical properties due to lactose fermentation. And on differential media, differential diagnostic media with lactose, like McConkey's medium, Endo is used in Russia, Escherichia form colonies stained in the color of the indicator due to fermentation of lactose. Memorize, Escherichia are lactose positive bacteria. And this media before inoculation, I'm repeating, they content of nutrient agar plus lactose plus indicator and plus alkaline. Indicators are different, and dyes are used like indicators. In McConkey medium, in McConkey's agar neutral red, in Endo it is fuchsin. And before inoculation of bacteria, this media have no color of indicators because of, did you notice pH is alkaline? And If pH is, memorize this very important thing for diagnostics of disease caused by Enterobacteria. If pH is alkaline, indicator has no any color. And after inoculation of the specimen, if there are bacteria, those are able to ferment lactose, and they are Escherichia, if we are talking about Escherichia, Salmonella, Shigella, and Escherichia are able to ferment lactose. Bacteria, I'm going to remind you, bacteria ferment sugars till acid products, and some bacteria ferment some sugars till acid products and gas production. So always acid products are developed, and Escherichia are able to ferment lactose. So due to acid products, pH is changed into the acid, and the indicator will get the color. So colonies of Escherichia on differential media have the color of indicator. Maybe a red color, maybe pink color, maybe a blue color on another medium. And it depends on the color of indicator. I'm repeating. Escherichia don't produce pigments. The color of the colonies of Escherichia on lactose diagnostic media depends on the color of indicator. Non-lactose fermenting bacteria, they are named lactose negative, and they are Salmonella and Shigella, form colorless colonies on this media, on lactose, on differential media with lactose. So on McConkie medium, pink colonies, what, like that, for example, they have color of indicator. And it is possible to differentiate bacteria by the color of colonies. And do you see colonies with the color of indicator? And they are formed by Escherichia due to lactose fermentation. And colonies without the color of indicator, colorless colonies, they are because of they are developed by lactose negative bacteria. If bacteria don't ferment lactose, pH is not changed, and colonies have no color of indicator. The Salmonella are lactose negative bacteria, and memorize Shigella are lactose negative bacteria. Why we are talking about these three genera, Escherichia, Salmonella, and Shigella? Because of very often from the patients with acute intestinal tract infection, very often either Escherichia, Salmonella, or Shigella are isolated very often. And really, bacteria from another genera, Klebsiella, Yersinia, Proteus, Gaffnia, Serratia, Moraxella, there are many genera. And so very important to differentiate, Escherichia from Salmonella and Shigella. So biochemical properties, biochemical properties of Escherichia coli. They are biochemically active, and they are able to ferment many sugars, including lactose, very important, don't forget, glucose, they are able to ferment glucose till gas production and other in the sugars, except sucrose. So they don't ferment sucrose, they are sucrose negative, memorize. And for identification of bacteria of Enterobacteriaceae family, very important to detect proteolytic enzymes. Proteolytic enzymes are detected on nutrient broth. And if bacteria, nutrient broth, or maybe mid-peptone broth, maybe just 1% peptone water. Peptone is a product of proteins destruction, and if bacteria have proteolytic enzymes, they are able to destroy peptone till gases production. Gases, we are going to detect gases such as H2S and indole. And Escherichia coli have proteolytic enzymes, and it is possible to detect these enzymes due to indole production. H2S is not produced. Salmonella, opposite, they are able to produce H2S, they don't. Indole is not produced due to proteolytic enzymes action, so it's important for identification of bacteria. Virulence factors. Realize that I did remind you that Escherichia coli belong to normal microbial flora. Now we are talking about strains of Escherichia. Those are able to cause very severe diarrhea that leads to lethal outcome in small children. So realize why Escherichia coli are so different, because of strains. We are talking about bacteria, those belong to one species, Escherichia coli. But strains that belong to this species, Escherichia coli, differ by virulence. Some of them are conditionally pathogenic strains, so they are able to exist in large intestine, like normal microbial flora. I told you anyway, if they spread by bloodstream to internal organs, they are able to cause infection if they have opportunity. So such infectious diseases caused by conditional pathogenic microbes are named opportunistic diseases. And it is kind of endogenic infection, because this person was not infected from outside. Disease was developed due to own microbial flora, endogenic infection, opportunistic infection. And they, at the same time, between human beings, strains are circulated of Escherichia coli. Those have very severe pathogenicity factors. So they are pathogenic strains. They don't belong to normal microbial flora. If any pathogenic strain enters human body, any parts of body, including digestive tract, these strains are able to cause escherichiosis, or they have very severe pathogenicity factors. But conditionally pathogenic strains, it is repeated, live in human intestines, normal microbial flora, but getting into other organs can cause urinary tract infections, intra-abdominal infections, even septicemia. Septicemia is a kind, or it is clinical form, when bacteria are able to multiply in the blood. If bacteria are transmitted by blood, just temporary, maybe a few days, there are many defensive factors in the blood, and if bacteria go to the bloodstream, they are able to survive just a few days, and then they are eliminated from the blood because there are many defensive factors, including macrophages, including antibodies, and such clinical form is named just bacteremia. Bacteremia means temporary circulation of bacteria in blood. But sometimes, if immune system is weak, if patient is immunocompromised, has any kind of immunodeficiency, bacteria, those are circulated in blood, are not eliminated from the blood, and they have ability to multiply in the blood, and such clinical form is named septicemia. It is very dangerous situation, and sometimes people die of septicemia, sometimes not possible to save the life of patient. It means immune system is weak, and of course any chemicals, any preparations are used antibiotics, but it is just 50% of affect immune system should participate in elimination of bacteria. And if patient has immunodeficiency, chemicals, medicines are not able to help. Pathogenic strains are divided into pathogens of parenteral. Parenteral means avoid digestive tract escherichiosis, and enteral escherichiosis. And escherichia, those are able to cause enteral escherichiosis, are named diarrheogenic escherichia. They cause very severe diarrhea, like travel's diarrhea and like children's diarrhea. And small children are very sensitive, and in some cases, lethal outcome is developed due to diarrhea caused by pathogenic strains of Escherichia. Strains of Escherichia coli, now we are talking about factors of pathogenicity of diarrheogenic Escherichia. We will study them. Strains of Escherichia coli causing diarrhea are divided into 5 groups in accordance with pathogenicity factors. Like you should learn by heart these 5 groups. And what are they? Enterotoxigenic Escherichia coli, ET/EC, virulence factors are enterotoxins. Enterotoxins, you know, all exotoxins are proteins, are toxic for human beings. And exotoxins have organs affinity. Every exotoxin is able to destroy any organ or any system mainly. And exotoxins are classified in accordance with action. They are named. So if exotoxin acts on digestive tract, it is named enterotoxin. If, like in case of botulism, exotoxin is able to destroy synapses, acts on nervous system, it is named neurotoxin. Is, for example, diphtheria toxin is able to destroy tissues, it is named histotoxin. If exotoxin is of bacteria, many bacteria are able to produce exotoxins for destruction of red blood cells, and they are named hemolyzins. And so every exotoxin should be named and pay attention on test, on exam. Instead of writing just toxin, you should realize, is it endotoxin or maybe exotoxin? And if it is exotoxin, you should realize what organs and system this exotoxin is able to act, and you have to write the name. So in case of Escherichia coli, we are talking about enterotoxins. Those are able to act on digestive tract. Then the second group of pathogenic strains of Escherichia coli, entero-invasive Escherichia coli, abbreviation is EIEC. They penetrate and multiply in cells. The same property have Shigella. Shigella, causative agent of dysentery, are able to penetrate and multiply inside the cells in the wall of intestine. And so they are able to destroy the wall of intestine. Ulcers are developed, and it is due to development of these ulcers, particles of blood are detected in feces, and some Escherichia coli, those are able to multiply in cells. They cause dysentery-like diseases. Again, the patient will complain that there are some particles of blood in feces, which is a sign that their wall of intestine is destroyed. Then next group, enteropathogenic Escherichia coli, EPEC, adhesive and damage intestinal microvilli. Okay, then enterohemorrhagic Escherichia coli, EHEC, they are able to secrete cytotoxin. Cytotoxin is able to destroy cells, and it leads to bleeding again, hemorrhagic. And the fifth group is enteroaggregative Escherichia coli, EAEC, fast attach to the cell surface. So you should learn by heart these 5 groups. And what is the difference? So enteropathogenic Escherichia coli EPEC is an important cause of diarrhea in infants, especially in developing countries, EPEC. Now, what is the difference? When we are going to do diagnosis of diarrhea, of course, we are not looking for conditional pathogenic strains of Escherichia coli. We are looking for exactly pathogenic strains of Escherichia coli. And what properties are they differ by? We are not looking for any strains of Escherichia coli. We are looking for exactly pathogenic strains of Escherichia coli. And from the beginning, morphology and staining. They have similar morphology and similar staining. All of them are gram-negative rods. So they not differ by morphology and staining. Then cultivation. All strains are able to develop colonies on nutrient agar, S-type colonies without any color. So no differences. Then biochemical properties. They are not differ by biochemical properties. All of them are biochemically active. They are able to ferment many sugars, including lactose, and they have proteolytic enzymes. So again, not possible to differentiate them by biochemical properties. So memorize, it's very important, and you should answer on test, on exam, what is the difference. And the difference is number 1, strains of Escherichia coli causing of diarrhea. They are pathogenic strains, and they have certain virulence factors, pathogenicity factors. Conditional pathogenic strains have just endotoxin. They don't produce exotoxins. They are not able to cause the destruction of wall of intestine. So they have no factors of pathogenicity. But it is very complicated to detect factors, to detect factors of pathogenicity in practice. In science, it is possible to study them, but in practice, it takes time and it is expensive. So we know about them, but we are not available in practice in bacteriological laboratory to detect these factors of pathogenicity. But fortunately, pathogenic strains differ from conditional pathogenic strains by antigenic structure. And I know that you did study on laboratory classes. Anyway, it is educational to repeat again. I'm going to remind you antigens of bacteria. O antigen, somatic antigen, K antigen, capsule antigen. They have microcapsule Escherichia. And if bacterial cells have flagellum, one or maybe many flagella, flagella consists of specific protein and it is specific antigen, H antigen. And if bacteria are able to produce exotoxin, exotoxin is a protein, and again, it is specific antigen. So strains those belong to the species Escherichia. They have O antigen, they have K antigens, they have H antigens, and strains those are able to produce exotoxins. They have a protein antigen specific for exotoxins. And bacteria strains, those belong to Escherichia coli species, they are differ by O antigen. And they have different O antigens by numbers like O1, O2, O3, and so on. And approximately till 176, approximately 200s. All strains of Escherichia coli have approximately 200 O antigens. And K antigens approximately 100 and H antigens approximately 50. So easy for memorizing 200, 150. Why you have to know? Because of every strain, pathogenic strain of Escherichia coli has own antigenic formula. Like for example, Escherichia coli O26, K15, maybe H, maybe 12. This is antigenic formula. And in science, of course, all antigens are detected. An antigenic formula is written for the pathogenic strain. Again, in practice, in bacteriological laboratory, not possible to detect all antigens. It takes time, and all antigens should be detected for sure. And we know what scientists did detect, what all antigens belong to pathogenic strains. Approximately 50 all antigens belong to pathogenic strains, and they are known. For example, you should memorize O26, O55, O111, and O1, and so on. And diagnostic sera, monovalent diagnostic sera produced for detection of one of certain antigen. Diagnostic serum O26 contained antibodies for antigen O26. Diagnostic serum O55 contained antibodies for O55 only. So it is possible to detect antigens. And of course, in diagnosis, they produce their commercial diagnostic sera, and by using of diagnostic sera, it is possible to differentiate pathogenic strains from conditional pathogenic. And finally, all antigen should be detected, and biochemical properties, morphology, staining should be detected, and it is possible to identify Escherichia, and exactly all antigens should be written. So now I'm going to we have just 2 minutes. I'm going to make a break, and again, I will send the link leaders and we will continue after the break. So 10 minutes break. And after the break, I will continue. I will send the link again. Okay, так.